Well, good afternoon. We're excited that you decided to stop by. We just got a quick demonstration here. It's going to be about 15 minutes. We just want to show real quickly that we're going to extract some DNA. So my name's Dan Hansen. I'm a father, a farmer, and I work for DuPont Pioneer in Johnston, Iowa, on their visitor and stakeholder outreach team. So we host visitors from around the world, all different cultures and all different walks of life, to showcase the science behind these products. What we're going to do this afternoon, kind of like a cooking show, I've prepared a few items here for us, and we're real quickly going to showcase that we can extract the DNA from strawberries or bananas or anything else. And all these materials we actually purchased at Target just this morning. So very easy concept. And what we have here, starting out, we just have literally water from the drinking fountain. We've got just a pinch of salt, about a teaspoon of salt, and some shampoo. And what that is those are mixed together and they are made up this solution, and I've already gone ahead and taken out that solution into 10 milliliters here. So what I need a volunteer. I got one right here. All right. So what I'm going to have you do is take a strawberry, cut the top part off like you were going to eat it, and maybe do that for a couple of them. And I'm going to ask you to set them in there.
Oh, okay. How many? 3?
Just 3 is good. All right. Just throw those back in there for me. And what I'm going to have you do is I'm going to have you start mashing those up.
Oh.
Go ahead and zip that bag up. And so if you think about extracting DNA, all life on the planet is made up of DNA, whether you're a plant, an animal, bacteria, a fungus, or anything else. All living things are DNA. And we're going to extract the DNA from this strawberry. Now, the first thing we have to do is break through the thing that surrounds every cell. Now, I know some of us have to think a little harder than others, but you think back to 7th grade science, and there was a cell membrane or cell wall that surrounded every cell, right? That cell has to be broken. It has to be crunched. So what she's doing here is just crunching, smashing up this strawberry. And if you got it nice and good. All right, we're gonna go ahead and open that up, and we'll have you throw these on for me.
Ooh, okay.
Make it look legit.
Yeah.
All right. Now open that up. We're gonna go ahead and pour this solution. Again, this solution was literally shampoo, a little bit of salt, and some water. All right, and I'm gonna have you go ahead and zip that back up and just put it down on the table and kind of roll it. So try not to create a lot of bubbles.
Okay.
Just kind of massage, kind of like kneading bread. And she's going to do this for about 60 to 90 seconds, and it's just going to ensure that this liquid is going throughout the bag and is making sure that all that DNA within the strawberry is exposed. What we will do very shortly is transfer that, and this is a scientific filter. Again, using a coffee filter and a rubber band, we are going to have her pour this material into here, and the liquid will come down to the bottom side. All right. You think you got it?
I think so.
Are you confident?
I feel
all right.
about it.
Just making sure.
Okay.
All right. So we're going to go ahead. We are going to cut the corner of this bag off, and I'm going to ask you to pour that slowly into the cup.
Oh, I made so many bubbles. I'm sorry.
That's all right. So pour the majority of it. That should be good. You don't need to have literally all of it.
Okay.
Hopefully you get a good enough job kneading that together that I won't need all of it. So we should be good. So thank you. You can go ahead and have a seat. Let's give her a round. Just do it. That's a good job. All right. So what we need to do now is just wait a couple of minutes for this to settle. And you can see it's starting to drip. It's going to be pretty slow. It is a coffee filter. It's not a screen of any type, so it's going to leak pretty slow. So after a couple of minutes, that material, again, just strawberries, shampoo, salt, and water, will allow that to come out. And then what we will do is take a little bit of that material and we will put it inside this vial. And after a couple of more minutes, we will then be able to, you'll see the DNA literally start to separate from that material. Now this would be the same if we did this with a plant, with an animal, with bacteria. It doesn't matter. I could literally have had a guest or an individual, and if you want to stay around after the demonstration, we can do it again. I'll show you it works. You could spit in a cup and we could extract your DNA. It would be the same thing. Okay? It would be literally the same thing. If you take a glass of the water, instead of having you gargle this, I'll probably have you just gargle water, and then we'll put the salt and shampoo in after that. But you could literally extract your own DNA. My training is as an educator, and when I was doing my student teaching, I did this with 7th grade students in a classroom. So it was very, very easy to do. I did have one kid that didn't gargle the water; he just drank it, so I had to get him more. But other than that, it's really foolproof. Other than when they don't follow the directions. So we'll see how we're coming here. We do need a little bit amount of that, so we'll wait just a couple of more minutes here. I'm going to try and get about 2 to 3 milliliters of that solution, so we're still waiting just a little bit for that to get through before we put it on this material. And while we're waiting for that, I should describe this is literally just rubbing alcohol. So this is just rubbing alcohol that we've chilled in this ice, and that's going to help the chemical process to actually separate the DNA and denature the DNA. So it's really a common process that many of you might even have done this in 7th grade science or in high schools or colleges to extract that DNA. Obviously, the biggest component, and when you really start to get into the deep science, is after you've extracted the DNA, how you analyze that DNA. But what we're doing for today's purposes, obviously, is just the first initial step to extract that DNA. So we'll see how we're coming here. Slow is how we're coming. So, no. So, what questions do we have so far before we move on? Don't be shy.
What's the soap for?
What's the soap? So the soap has got, the soap simply has what's called, let me see, make sure I say that, the FDTA, and it's just a component of the soap that literally allows for the denaturing of the DNA, the separating of the DNA. So if you think about DNA, it's the double helix, right? So it's, you think about that twisted ladder. You guys remember that?
Charged to one side, not the other.
Yeah. So that twisted ladder was...
Soap acts like a surfactant, right?
What's that?
Soap acts like a surfactant.
Yeah, so it pulls that, pulls that apart. So when you think about that, we're essentially denaturing those sides now in a process, a common process. If you're familiar on the more scientific term, that polymer chain reaction, the PCR process, is actually where you're denaturing that DNA, making those copies, and then adding those together. But again, this is the very start. This is we'll keep it simple for today's purposes. As you can see, Target brand materials, so that's obviously going to be pretty simple. So what we're going to do now is we're going to see if we can't move this over. Kind of looks like a strawberry margarita. I'll let that keep going, but in the meantime, take we're going to let that sit here for just a second. So you don't want to shake it, but we'll just gently swirl this around. It's just got to sit for a minute or so. So as that's, what's that? So alcohol, again, is just similar to what's in it's just all the components, it's all the chemical, or not chemical, excuse me, it's all the components that allow for the separation to occur, but it's just household items that you could get instead of utilizing, items that you might utilize in a lab. Obviously, we're not buying baby shampoo to do it in the lab. So we should start to see, you see it forming there a little bit? What you should be able to see now is as you stir this around, that, ladies and gentlemen, is the strawberry DNA. Back a little bit. Sorry. It's my first gig. I've never been on TV, so. So that is the strawberry DNA. That DNA, again, is literally made up of 4 base pairs that make up all life on the planet, A, T, C, and G. And so what we can utilize that for, that knowledge of understanding that, is we can actually find certain sequences within plants that we want to ensure we breed those plants, because certain genetic sequences, that genotype, the physical characteristics, the genetic sequences that you can see, the genotype leads to something good for that plant. Just like genes, ATCG combination, lead to whether we have light hair or dark hair, or blue eyes or green eyes, we can find certain sequences that are going to help lead to a plant that is going to be more robust against a particular disease or grow better in a certain environment. And we can use those sequences to then go ahead and continue to separate out and figure we want to use these plants and we don't want to use these plants. Obviously, then we move into what most people consider a GMO today, and that is when we can find a certain sequence that's helpful and ensure that that is in that plant by moving that to another spot in the plant or moving it to another species of that plant. So making sure that we are utilizing those and figuring out which plants are going to be the most useful and most effective for farmers to grow in fields today. Well, please stick around if you have other questions, but thanks for attending, and hopefully you enjoyed seeing a little bit about DNA. You can try it at home.